Okay, right back into this. Now, this is kind of like a part 2.1 or a, or a 1.1, 1.2. Yeah, smoke another one. So, sciencey stuff though, really. A lot of things going on right now, and we have to be aware of all the science. Because science is speaking, and science, well, science is good. We all know science is awesome, and this guy is gonna break it down for us. And I'm gonna do my commentary through it, just because it might help give people a different perspective, or I guess a different, more in-depth reason about what he's talking about and why it matters so much right now at this time. So hopefully I can get one of these videos that are streamlined, because damn, there's a lot of information. I can't record it and upload it because it just takes too long. So I just do it the live way, and it'll come out eventually. Watch them all. It's a series. It's a collector's set trilogy. So anyway, here we go right into it, and he's going to give us a breakdown about how the coronavirus is infecting our cells, why it matters that it's infecting it this specifically, or this specific way. Some pretty cool stuff here.
Hi, I'm going to show you some of the molecules coronaviruses use to infect human cells. So I'm particularly interested in Wuhan coronavirus, which is the cause of the ongoing epidemic in China and is spreading around the world right now. Let's take a look at what a Wuhan coronavirus looks like in electron microscopy. Okay. All right, so I have an image of these black arrows show two of the Wuhan virus, two copies of the Wuhan virus. And you'll see that it's a round particle, and on the outside, it has these spike molecules. They're these little fuzzy guys around the perimeter on the...
I'm going to stop that right there and just say that the little spiky guys that he's pointing at there, the little fuzzy things that are protruding out of that little central dot, is those are basically where all these proteins are going to reside that are going to be able to infect us. It's going to come into play later on, but we'll save that for later. But just notice how plentiful they are around there, and we can see that, you know, this is the Wuhan coronavirus. This is the actual virus under a microscope, and it's got all kinds of little spiky things, right? And we'll see one later. Just take that for note.
The inside, there's RNA, which encode the instructions of the virus. And the virus's goal is to get inside your human cells in your lung, to dump its RNA into your cells so that your cells can produce more copies of the virus. This strategy is not specific to coronaviruses. A lot of familiar viruses use these molecules that form spikes on the surface to invade human cells. Let's take a look at a couple other examples of that.
Before we do that, let's just take a prerequisite course in how viruses infect our cells. And I hope I can find this. I hope.
So I just...
Yeah, back here.
55%.
Slow down.
It's about 75% identical, which is a high amount of identity.
Chill out there, George. Okay, so this is a very cool video about different viruses. There's different types. It's very informative. I hope you enjoy it. I hope
Icy hot. Rise from pain.
because it took a while to find this, but anyways, this guy goes into it. We're gonna skip a little bit of the blah 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 blahs, because we don't really need to know a whole lot of it, but we will start right. Well, how about yeah, there. Because viruses, let's just start at the point where viruses actually they're not alive. They don't have a process that keeps them alive. They need our cells in order to replicate. So let's just go into a little prerequisite course, and then we'll get back into that heavy stuff.
So viruses exist in a kind of gray area in between simple molecules and living organisms. So what's inside a virus exactly? In truth, a virus is pretty much just genetic material in a protein casing. There is no membrane, no organelles, not much of anything we are used to seeing in living creatures. Nevertheless, viruses reproduce by injecting their genetic material into a host cell, thereby hijacking the cellular machinery of the cell and forcing it to make copies of the virus instead of what the cell would normally be doing, sort of like pirates on the high seas, capturing a large vessel and taking command. Viruses were discovered in the late 19th century when examining certain diseases that afflicted plants. It was found that sap from the plant would transmit the disease, even though no bacteria were visible in the sap when examined with a microscope. And the sap would still transmit the disease, even when it was filtered by a process meant to remove any such bacteria. This meant that the agent responsible for transmitting the disease must be way smaller than a single bacterium. But this agent could not be cultivated in test tubes or petri dishes, so it must also be much simpler than a bacterium. Later, as we became able to examine viruses with more sophisticated techniques, we began to realize the structure of the virus, which comes in a number of forms. Some are rod-shaped or helical, like the tobacco mosaic virus. Some are icosahedral, like an adenovirus. Some have a membranous envelope covered with spikes, like the influenza virus. And some even look like weird little spiders. This is called a bacteriophage, and it's kind of like a rod-shaped and icosahedral virus combined, with some fiber tails. The thing they all have in common is that they carry their own genetic material, which could be double-stranded or single-stranded, and either DNA or RNA. This will typically be found as either a single linear molecule or a circular molecule. The protein shell that encloses the genetic material is called the capsid, which comes in different shapes for different viruses, and the capsid is made of smaller subunits called capsomeres. That's really all there is to the structure of a virus. So how exactly do they reproduce? As we said earlier, viruses hijack the machinery of a host cell. This is because they do not have ribosomes or the other components necessary to express genes, so they need a cell to do it for them. Certain viruses are able to infect certain kinds of cells, and this is due to the system of recognition that must occur between the two.
And right there, that's the whole. That determines how vicious a virus is, how infectious a virus is, what type of cells it can infect. Is about all with communication. As if the virus is basically it's hijacking the cell, it's tricking our cell. It's like a Trojan horse kind of thing coming up unsuspectingly, and the cell allows it come in and not recognizing that it's a threat. which is very sneaky. Okay, but there's different types of ways viruses can get in. We'll just run through this. We're not going to go through the whole thing, but just a little bit more, and then we'll jump right back to the other video. It's going to give us a little bit of a, build a path, so we can maybe make some sense out of this.
In order to get inside the cell, a virus must be recognized by surface receptors on the cell. So there must be some specificity for these receptors to that particular virion.
And again, we'll pause there for a second. And what he's talking about there is that there needs to be, like, viruses don't have the ability to move on their own. They just float around inert. Like, if you look in that picture in the left with the it's showing basically on a really small minuscule level, like that, this tiny little virus, it's actually a lot smaller than our cells. And if you look at the main image here, you see, like, that's a pretty good representation about how big a virus is. Like, it's tiny compared to, like, the cells that our cells that it's trying to infect, and the certain specific cells that this type of coronavirus is trying to infect is our T cells, our immune cells, which is highly weird. Okay, so anyway, I'll stop rambling, but it's the communication methods, right? And what can change the communication methods is these proteins that are expressed. When you see these little blue dots, those would be where, let's say, the virus's mechanism of communication would be, right? If you can understand that, and then the cells, the green ones, the receptors, right? They're being fooled, right? They're being fooled right now. They kind of bind on to it, and they're like, oh, well, there's nothing really wrong here, and then it brings it in a little bit closer, and it fuses with the cell, and it's able to inject the capsid, right? And then that's how it gets copies and infects. But what the main thing to take away here is the proteins and the communication and the surface envelope where the blue parts and the green parts are. So all this mechanisms can happen. Communication must take place, and if you can speak more languages, you can obviously communicate better, right? Right.
For this reason, many viruses are specific to only a small set of species, or even one individual species, and sometimes even a particular type of cell found within that individual species. Once this recognition occurs, the virus either injects its genetic material into the cell, if it's a bacteriophage, or the virus can be brought inside the cell completely intact through endocytosis. Once inside, the virus disassembles and the viral DNA gets transcribed and translated by all the parts of the cell that are typically busy working for the cell itself. Once there are many copies of the viral DNA, the capsid proteins reassemble and form new viral particles, up to hundreds or even thousands of them, which then exit the cell. Sometimes this process can damage or destroy the host cell. So let's look at the different mechanisms viral replication can utilize for bacteriophages.
Okay, we're not going to go any further than that. That's just the replication mechanisms, which isn't so important right now on the topic I'm trying to follow on. But it is. We all know viruses can replicate pretty fast. So back to this, and he was saying how it infects us. Different ones infect us differently. This here specific virus is going to infect us in the same way. It's going to trick our cells like a Trojan horse. It's going to come up and it's going to fuse with our cells. It infects us the exact same way as the human immunodeficiency virus, HIV. It infects the same way. It's not HIV. You know, it's you know what I mean. Like if you took tires off of one car and put them on another car, it doesn't change the model of the car. You know, you just put different, uh, you know, tires, different parts on it, better handling parts, let's say. Um, so yeah, this specific, and this is coronavirus that we're looking at right here, the one that is the Wuhan coronavirus or COVID-19, and yes, it's a lot, and I'll try to break down the science for you as best as I can from where I see that there's things that he's hinting, and there's also numerous papers. Or actually, I shouldn't say numerous. There's two. There's two, but they are papers, and we will get into them after about the scientific papers. And this guy, actually, yeah, there is numerous because this guy, there's three, but I can count. So back into the video, and hopefully my commentary gives some insight instead of some excite.
This is an influenza virus example, and in the center is an influenza virus particle, and you see again the spikes, the little fuzziness along the outside. The two other things are just empty membranes. They're there to let researchers see how does the virus actually fuse with these membranes. So the idea is how does the virus fuse with the actual cell, but this is a simpler model of that. And we can see I can kind of slice through at different depths and at different angles. And if I get the right position, I can see where the virus is contacting this nearby membrane.
Okay, I paused that just perfectly. Just to you can see where the blue cone is he's pointing to, and there's like an opening. You know, it's like you can see it's a continuous circle, and then there's a little opening there. That is the process of fusion happening with this specific cell, right? It's allowing the cell to bind, and through a process, you know, there's endocytosis and fusion. But basically, what it's doing is it's allowing the virus to inject its material, right? That's how the process is kind of happens, right? But this specific strain is influenza. It's not coronavirus. The one just before this one was coronavirus. But it has the same kind of outlook, right? It's all got all the little spikes on the outside, and the last one we looked at, coronavirus, it had multiple spikes on the outside. It's quite a bit, right? But there's some unique characteristics here. Let them keep going.
It's formed this little cusp. It's going to fuse with it and create a hole so that the viral RNA, the genome, can be transferred into a cell. Let's look at one more virus that uses the same strategy of spike molecules on the surface. We'll look at HIV virus.
That's huge news, right? Okay, I said I wasn't going to pause it again, but I did. Let's look at HIV virus. Okay, he's doing this, right? This is research on coronavirus. Like, why is he just pulling HIV virus out of all the tens of thousands of viruses he could choose from? What's so significant about HIV virus in this specific study? Curious? How come it's letting me not do that? Okay, there we go. Spike protein. Okay. HIV-1. Right? That's the virus. But now this here is a protein. Glycoprotein. This is on the spike protein. This is on this. in a virus. All viruses have spike proteins, most of them, certain ones. But like especially the coronaviruses and the influenza, they have spike proteins. They have all these little fuzzy things that surround them. They got the little, you can see the ball there that's rolling around. It's got the little fuzzy spikes all around the outside of it, right? That's where this protein is located. And what's specific to this protein is that it's only found on HIV 1. And actually, 2003 SARS outbreak came out of China. But there are differences this time. Notable differences and differences that they say, hey, this is a lot. But the proteins are what we're trying to look at here. We're going to go back into the science. We're not going to go into too much speculation, but we're going to definitely point some elbows. Yeah, because it has to be done, right? It's only logical. Really, it is. Like, you can't not think of this right now, and if you do, I'm sorry, or if you don't want to, well, then just go about your thing and do what you want to do. But I hope somebody gets something out of this. And he is now going to talk about the HIV virus and how it infects a cell. And what's so special about that he has to throw this in here, a presentation with the current Wuhan coronavirus. Strange.
So here are a half dozen HIV virus particles. One thing you'll notice difference, we see the membrane around the virus. It's spherical. Again, I'm cutting through it, and I can sort of slice in and out through the virus. And one thing we'll notice is that there are many fewer spikes. That influenza virus and the coronavirus were covered with the spikes. HIV virus is a little bit different. It only has tens of spikes. For instance, here's one, this little stub sticking out. Okay, so we're going to look at these spike molecules. They're used by the virus to recognize your cells and to enter your human cells in your lungs, the coronaviruses in your lungs. We're going to look specifically at the spikes on coronaviruses.
And he's kind of like saying a little bit right there, right? The little fumbling of the word. He didn't fumble that by accident. There's a little certain thing that he's specifically trying to say without saying it. He's a scientist, and this is from, he could get in big trouble if he starts pointing fingers. But he's just laying it out there. And what he's talking about was what's so important about why he said, look at these HIV viruses. Now you see how it's like a big line that's going right through that whole thing. It's like encapsulating these cells, those little smaller circles. It's like holding them in. It's kind of like a curved, right? Like the cells are trying to rise up to get out. Well, that's exactly what they're doing. And there's a reason why he showed it like that. Because this infects us. It doesn't infect us like any other coronavirus. It infects us like there's a different way that cells or viruses replicate. And in this way, it takes over the cell and then basically it's a T cell. It's a white T cell. It uses the CD4 receptor, so that makes it a T cell. It's basically connecting with those cells specifically. But anyways, yeah, it's a lot of science, and I still don't know a whole lot. I will guarantee you that it's like, but there's like huge connections here as to why he's saying, this is the way it is. And we've seen the past coronaviruses how many spikes they had, right? And look at how remember he pointed here and said, like, to look at this just little one spike. There's only very few spikes around these little small circles. but it's still highly, highly, highly infectious. Right? It's HIV. That's HIV, what we're looking at. Exact thing. But what makes it so infectious is the proteins. This glycoprotein that allows it to infect your cell. A virus is useless unless it can actually infect your cell. And for some reason, the infectivity, or the things, the glycoproteins on these spikes from HIV somehow found their way onto this new virus. Really weird. So let's go back. And I'll try to keep the commentary as on point as possible.
Okay, so here I've brought up some electron microscopy of human coronavirus, and it's looking kind of pale. Let me try hiding this and showing it one more time. Let me try to make the threshold. Okay, I just changed the threshold of this electron microscopy, the displayed threshold. So this is what one of these spikes looks like on a coronavirus. Okay, it's a human coronavirus. These human coronaviruses, there are 4 different strains that are known. They cause colds. It's a mild illness. They cause about 10% of the common cold, cases of the common cold. The virus would be here at the bottom, and this is the spike sticking up. So this is the top here is the furthest part away from the center of the virus. And this is what the electron microscopy looks. This more detailed view than what we were looking at a second ago was obtained by averaging images from like 10,000 different spike molecules to get a higher level of detail.
Okay, and remember, this is a spike molecule that he's talking about, right? So it's not the virus itself. He's giving a demonstration about the spike molecules for a specific reason. And he threw HIV in here for a specific reason. Because it does have the HIV protein. And the reason why he's targeting the molecules is he's basically coming out and affirming this paper. Exactly what this paper points at. Right? These proteins, these glycoproteins, and there's a reason why they're like this. Right? And they're cleaved into subunits. S1 and S2. They're 2 domains, and they're receptor binding sites. Receptor binding sites are where these places are able to interact and the communication can happen so that our cells will let the virus in unknowingly, unwittingly. It's like, it's like a big... It's crazy. It is. So... and they said, hey, these don't look right in here. They do not look right. And they did a sequence alignment, and it matched nothing. And everybody shot this test down, or paper down, and said, no, it's not true, it's not real. What it actually is, yeah. It's a lot. It is. And what they say here is really interesting because it affirms what he's going to go into later. And it says the novel inserts are part of the receptor binding site. Okay? The inserts are part of the binding sites. Okay? So that makes it, that's how it's going to infect us. So there was something done to these sites to make it more infectious. So that it could recognize our cells and get in our cells easier without detection. And actually, very efficiently. And it's so hard to detect because this is actually hitting our T cells. Because it's a CD4 reception that is taking place. So basically, when there's only certain cells that are CD4 that have this CD4 expression. Right? CD4 is basically a protein or a receptor protein or a binding molecule that's going to accept the glycoprotein molecule which would be the proteins of the HIV virus, which makes it so infectious. So they took these little pieces off that were the infectious pieces of the virus, and they stuck them on whatever the hell kind of fucking Dr. Jekyll, Mr. Hyde shit is going on under there. Because it's not just a regular coronavirus going on under there either. No, it's not. So, what he's going to affirm here in this later video that we're going to see, they say in this paper, we speculate that these insertions provide additional flexibility to the glycoprotein binding site. And why does that matter? Why does it need to flex? And it's got a hydrophilic loop. And that's basically means hydrophilic means that it can repel water. Hydrophilic. And philic meaning like to be like repel or keep away. Like hydro water. So it's keeping this hydrophilic loop. And it's gonna be very hard. Very easy for this interaction to all take place on a chemical level. It's a lot. And I'm trying to, but yes. So he goes on to this video. We're going to get back to the video. But what's super important here is he's going to affirm later, and I'm not sure where it is in the video, but he does say it. I'm giving you this as I just literally learned about this like 4 hours ago, and I'm trying to streamline it as best as I can. So yeah, it's going to provide additional flexibility to the glycoprotein binding site. Okay, and we're going to see why. These binding sites, and then this is going to show up later too. Let's go back to the video. Science matters. This is real science.
And from this electron microscopy, we can figure out exactly what the molecular structure of the spike is. So let me show you that molecular structure.
I'm going for a cigarette. I'll be back. I've seen this.
Okay. So here's what the spike looks like. And we have 3 proteins here. There's a green one, a blue one, and a red one. And they're actually all the same. They're 3 copies of the same one, and I colored them differently just so that we can clearly see the 3. The spike is symmetrical. Okay, all 3 copies are more or less identical in their shape. The orange part I'm going to talk about a little bit later are sugars covering the spike, sugar molecules. And the really interesting part of this structure is the molecule that the virus uses to get into the cell is actually here. And it's this little tiny thing. It's a sialic acid molecule. And it fits in this little cavity in the spike. The little blue dashed lines, let's see if I give you a better bet. There's some little blue dashed lines. Those are hydrogen bonds. Those are attractive interactions between the sialic acid and the spike. Okay. When the virus encounters one of your cells, and your lung cells and other cells in your body are covered with these sialic acid molecules, it knows it's found your cell because the sialic acid molecule sticks here and triggers the initiation of this reaction, which is going to make the virus invade the cell. All right. So that's a human coronavirus that causes colds, not the Wuhan strain. Let's take a little closer look first at this sialic acid. It's a very small molecule. I just want to briefly show you that. Okay, it's this little speck here, only a few tens of atoms. It's a sugar, it's a monosaccharide. And you might think, I guess another important fact, influenza virus invades your cells and recognizes your cells by the same molecule, sialic acid molecule. Okay, so that's interesting. You might think that other coronaviruses, well, they're in the same family as this human coronavirus, would also use sialic acid, but surprisingly, that's not the case. And so let me show you what the Wuhan coronavirus is recognizing, and also SARS, a virus that caused an epidemic in 2003, what it recognized. Oops. Okay, so let me try to get it. Where is it? Here it is. So Wuhan virus and SARS virus are recognizing this protein, which is, again, it's a natural protein in your body, like this sialic acid on the surface of the cells. This is called angiotensin converting enzyme 2, ACE2. There's also an angiotensin converting enzyme without the 2. Okay, this one with the 2 lowers your blood pressure. The one without the 2 raises your blood pressure. Both of them work. They have this groove. Let's see if we make this groove bigger. This groove here. Right in here. Both of them work by clipping a small angiotensin molecule. It's 10 amino acids long. The ACE without the 2 clips it from length 10 amino acids, angiotensin from 10 to 8, and that raises your blood pressure. And this ACE2 clips it from 8 to 7, and that lowers your blood pressure. So this is actually a good molecule. Both ACE and ACE2 are interesting targets for a lot of diseases, for high blood pressure, for diabetes, for heart disease. At any rate, we're not going to be able to avoid Wuhan coronavirus by getting rid of this. This is an important, good molecule on the surface of your cells. So how does the virus spike recognize this molecule? Let me bring up, I'd like to show you the Wuhan coronavirus, but we don't have a structure of it. So I'm going to show you SARS, which is the closest thing we have, the spike structure.
And this is going to be very important coming up. Remember I was saying, where was it? How it's going to provide additional flexibility to the glycoprotein binding site. And I know that might not just sound like anything, just a bunch of blah, blah, blah. That's everything. Fix my blankets. Oh, my makeshift streaming site. But it's just to get information out to people. Cause I'm just coming up with this. I'm figuring it out on the fly too, right? I'm just a regular guy on the fly. So I do know a little bit about a little bit, but not to the point where I can make a definite conclusion on my own, but these guys are doing it for us. Right? We just gotta put the pieces together and understand it. So, this is the paper that everybody freaked out about, saying that it was a nuclear weapon or bioweapon. Or, not nuclear weapon, sorry. A bioweapon. This is the paper. And let's just go back into the video and remember about the additional flexibility in the glycoprotein binding site. Coming up.
Structure of SARS.
And he's given us a structure of SARS here, because this is the closest relative to the current, virus going around now. The Wuhan coronavirus, or this COVID-19, or SARS-2, or nCoV. There's so many names. The Wu flu, yeah. There's a lot, right? But anyway, it's heavy stuff, and it's a lot to take in. I understand. But, we got Google at our fingertips. We are the people. They're not going to tell us. And they're not telling us. They have no information yet. Bullshit. This guy's got it figured out, and he's got a video. Like, I found this video within, I don't know, I just somehow stumble upon this stuff, and it's just, I think it's good fortune, or whatever you want to call it, but yeah, it's here, and now we're gonna observe it. So, flexibility in the glycoprotein binding site. SARS is the closest relative to this. That's why he's comparing it. But there is differences. Meaning that the SARS 2003 outbreak I don't know if we all remember that. It stopped. It was in China. Came out of there. A lot of people think that that came out of the lab. There's quite a bit of, but nobody can prove it, right? But this may prove that, hey, if it did come out of the lab back then, now this is just a different version of this. It's kind of a little different. SARS came from the bats. Remember, it was the bat protein. This has got the bat protein in it as well, right? But this has got the glycoprotein, which makes it more infectious, and there's other things going on. Like he says, he doesn't have the 3D model of it, of the coronavirus going around now, but SARS is the closest one. Now, back into it. Okay. Glycoprotein.
And this is going to look like a lot like that human coronavirus. Still, the three proteins. The sugars aren't here, but they aren't shown here. But in fact, there are sugars here. It was just in this particular model, they didn't add the sugars. Those sugars are part of all the spikes, the coronavirus spikes. And what they saw when they looked at SARS is that these, at the top of the spike, the virus would flip out part of it would make a small motion. Let me show you what that motion looks like if I can. So let's see, here we go. Okay, do you see this little green part at the top? It's called the binding domain. And it can move from a down position where all three of them are down and kind of tucked in to an up position. And that up position is the only position that can recognize this ACE2 molecule.
Okay, we're going to stop right there for a second. And what I was saying earlier about the flexibility of the binding site in the paper that everybody trashed, they go on to say, hey, we speculate that the insertions provide additional flexibility to the glycoprotein binding site. Why would additional flexibility matter in a binding site of a virus? Well, it would enhance its infectability. It would make it more infectious if you were able to, enhance it, right? And that's what they're saying here. Everybody's saying it. I've been saying it, but now, the science is only coming out, and goddamn, right, like, power to the people, because we gotta fucking talk about this shit. Nobody's talking about it, and I'm not a scientist, and I don't understand this, but I'm trying to learn it for you because I know there's something wrong. And even if it helps one person to say, hey, fuck, I got, what the hell, I can go spend a couple of weeks at the cabin, or I can go do this, or I can go, fuck. I'm not saying to do it, but I've taken my precautions, and I firmly believe that this is not something we should be trusting at all. This isn't like any other outbreak. This isn't like anything we've ever imagined. This is different. And this is why it's different. Not natural. It's not natural. There's no way. And this is what the science is saying as well, right? So they say it's going to provide additional flexibility, right? So it's going to facilitate or enhance virus-host interactions. Virus is the bad guy. Host is what it wants to infect. Anything else. And they're not only saying that it can only enhance infectability in humans. This might infect multiple hosts. This could infect birds. This could infect animals. This could infect... We don't know. But it sure seems like it has that high probability to do that, considering that these proteins are somehow super fucking proteins now. Like, it shouldn't happen in nature. Nature doesn't do this. Nature follows an evolutionary curve. They did they did the they did the genealogy evolution, and this is legit. And it, you know what I mean? It follows a little path, and its closest relative is the last SARS virus in 2003. But there's something very unique about this one this time. Meaning that these proteins couldn't have evolved from 2003 till now. Like, it would have took thousands, or if not tens of thousands, hundreds of thousands of years to collect that type of capability, that it could take something from the HIV virus, those specific proteins that are specific to only that virus, and somehow incorporate them on this new Wuhan virus. Like, it just, like, it's the past SARS virus, but it's just, anyway. The science is here. It is. And maybe you don't understand this, maybe you do understand it. I don't know how much time we have. Honestly. And I'm not trying to be a scare or fucking fearmonger or anything like that. I'm just trying to give people a heads up. And if I'm wrong, I'm wrong. You know what I mean? And I'm not, you know, you gotta make your own assumptions about this. I've made my choices. Right? I have. I'm going to show you why I've made my choices the way I did. And if I'm wrong, fuck, I hope I'm wrong. I don't want to be right. But there's really high reason to believe, and the science proves it, that this is somehow engineered. This is made this way. It doesn't happen by coincidence in nature like this. Maybe over a long evolutionary timeline. But not even in the timeline of recent humanity. No. Doesn't happen. Evolution follows a curve. Evolution doesn't spike. You know what I mean? We didn't go from single-cell amoebas floating around in the ocean to fucking multicellular fucking conscious beings overnight. It just didn't happen. On a timeline of time, you know, in the biggest universe, well, cause I'm not just going to spiral now, but anyway. Everything we've known on this planet is just literally like a flash in the pan on the scale of things. Really? Okay, so back into science. Let's try not to get sidetracked.
Angiotensin converting enzyme 2.
Okay, and ACE2. Why is that important? Well, ACE2. Okay, the ACE2 receptor is specific also to our T cells. So this thing is actually binding to our, it's weakening our immune system. But it hides and infects us like the HIV virus, if that makes any sense. And we see people getting better and getting released from hospital. Well, the HIV virus is known to, like, I guess, go into like a state of remission, and then something activates it, and it activates up again. Right? But it's a lot of confusion. It is, but it has those general properties around it. Even the virus itself, its behavior is showing highly transmissible infective properties that nothing else has ever had. And here we have scientific proof that says, hey, you know, this is a virus that shouldn't even exist, really, by nature, but here it is. Glycoproteins, binding sites. That's what we're looking at. The spike protein here in how a virus infects your cell. It uses this spike protein, and what's critical about this specific protein is that on the coronavirus, it has the HIV glycoproteins. Not the HIV itself, just a part of it. The infectious part. The important part. Because without that part, the virus can't communicate with our cells to get in. So this just gives it a fucking master key to get in wherever it wants. And essentially. So, on with the show.
When they saw this in electron microscopy, they only would see one up at a time. Like the red, blue, and green are identical, but only one would be up at a time. They didn't see two up, they didn't see three up. They just sometimes see one up, sometimes see it down. Okay. When it's up, they also saw it bound to this ACE2. And it doesn't bind to just any old place on the structure. It's very specific on where it wants to stick to the ACE2. So let me show you exactly the correct location where it sticks. Okay, so here's the actually from the electron microscopy that shows the ACE2 in brown. Here's the spike, it's flipped up. When it's attached to the ACE2, what they see in the electron microscope is the thing is even more flexible. So let's see. There we go. Let me see if I give you a better angle that shows this motion.
And that's what he just said, remember? This seems like it's even more flexible at this binding site. Right? And this is where the binding site is from this paper. They even mention it in this study that points that this here is not, in fact, natural. Where? Somewhere. Yeah, it's crazy. Yeah, and glycoprotein 120 plays a high crucial role in recognizing the host cell by binding to the primary receptor CD4. This binding induces structural rearrangements in the glycoprotein 120, creating high affinity binding site. High affinity. So very infectious, really. Right? Got a very good chance to be more infectious. That's what they're pointing at here. That's what they're saying. And this, when this receptor is up, right? When it's up, it can search, right? But when it's down, it's like it can't even be detected by our immune system. It's super stealthy. So let's just get back into this. This is the SARS coronavirus in 2003 that caused the big pandemic. Its structural genome compared to the Wuhan coronavirus to date, but the differences are, specifically in the spike protein, these glyco 120 protein inserts. So yeah.
Okay, so the thing can swing in and out, and what they observe next is that half of the spike just falls off when the ACE2 attaches. This is in SARS. Okay, what the top half falls off. And here I changed the color scheme on you a little bit. The bottom half I colored light colors, light red and light blue, and this light green kind of looks gray. Each of these three copies of the protein is cut at a certain point, and I marked it in red where it's cut. A protease, another molecule, makes a cut right here. And that's totally critical. If the cut's not made, then the virus can't infect. Okay, so each of the three ones has a cutting point, and what they observe is this green copy, the top half with the ACE2 floats away, and then the red and the blue, they don't have ACE2, only one of them. These become unstable. They no longer stay attached and they float away, and we're left with just this bottom light-colored portion of the spike. And this bottom portion in the middle exposes the mechanism which allows it to fuse with the human cell.
And right there, what he means by that is this whole process up until this point was basically just like a handshake between the virus and the cell, right? They're just communicating. And the virus is like, hey, I'm just here to just check on your hot water tank, anything, right? And then the fucking, the receptor cell, our cell, doesn't recognize it as a threat. So it starts, this process that allows it to basically expose this mechanism where it's going to be able to fuse with the cell. It's opening up the gates basically and allowing whoever, like, to come in, right? And that's what's happening up to this point. Roll.
Okay. Is this what's going on in Wuhan coronavirus? So everything I'm showing you is for SARS. We don't have the structures for Wuhan coronavirus, but one thing we can do is we can look at the difference between this SARS virus and Wuhan virus, because we know the chemical structure of the Wuhan virus from its genetic code. So let me color the differences between the two. Let's see, I have a little button I made to do that. There we go. So I colored every part of this green molecule, 1/3 of the spike, orange if it was different between the Wuhan and the SARS virus. So you see, it looks like a lot of orange. It's only about 25%. It's about 75% identical, which is a high amount of identity. When two proteins in your body are 75% identical, generally they function nearly exactly the same. Okay, but it would be what we'd really want to look at here is sort of the really important part here where it's attaching to this ACE2 molecule. We see a lot of orange at the interface between the two. And that would make me suspect that this isn't quite right. Wuhan virus. SARS virus and Wuhan virus are probably a bit different here. And in fact, if I only had this evidence, I would probably think that Wuhan virus wasn't even using this ACE2. Okay, because there are so many differences right there. But experiments that were done in the last few weeks already confirmed that Wuhan virus indeed is using ACE2. Okay, but we can expect that our model based on SARS is going to be a little bit off because there are a lot of differences in the critical location where it binds. All right, I want to show you one last thing, and that is what can your immune system do to fend off this virus?
So that's big, right? Okay, you're looking at like this orange space. That's the differences, right? And the top part, like the very top gray mass or brown mass, however you want to look at it, that's the ACE2 binding site. And right where it changes color and it goes to like the green and the orange, right in there is the binding site. And what's really important about that, he doesn't really come out to say it, but I know though it might not seem like that's very important, and some people might not get it, and I don't completely get it at all, but I do get it a little bit, enough to understand that it means a fuck ton. Because it changes, it's yeah. And he even said, that makes me, suspicious, something. And he's like, just given the data that I had, I wouldn't even know it was using the ACE2 cell, right? So this is, you gotta think that this is very hard to do science wise. And not only that, like, they do have fantastic tools to do this, but you gotta know where to look. It's like the biggest proverbial needle in the haystack you could have ever imagined. But we've mapped our genomes and certain viruses that we already know, and everything's in databases, and we can reference things and we can run it through. And what we found whenever we ran this virus through was it was nothing like anything in our database except the SARS 2003 virus, which, this is similar to, and which meaning that it came from a bat, right? It did come from a bat, but a lot of people are like, well, it came from the market, and come from, given the way this is, the structure is, it's not fitting that this specific protein would find its way on this glycoprotein 120 from the HIV virus would find its way on to a coronavirus or a SARS, like the SARS virus. It's so infectious. Basically, you have the influenza flu. It's traveling like a very, very infectious flu. It's way more infectious than flu. It's more powerful. It's spreading faster, way more faster. Just look at the cruise ship, right? We like something went down on there and they aborted that mission real quick because everybody got infected. They didn't realize how bad it was because China was just doctor in numbers, and the cruise ship was where the turnaround point happened. Right? They got data out of the cruise ship and they were just like, oh, holy shit. And that's why they did such an about face, and the US went and evacuated their citizens right away. Everybody went and got to people because Japan just said, guess what? Come get them, or we're, we can't deal with it. We got our own problems. Really, they did. That's basically how it went down. Come get your people. We can't do this because we can barely keep our own people going. So yeah, it became extremely infectious. So what we have is this virus that spreads like the flu, but infects us like HIV. It's not HIV. Don't get that mixed up. It's not HIV. It's like, it's in a class of its own, but it just infects us. Same method. And that's what they're talking about here in these glycoprotein structures. It's about why it's so unique. Right? And then they go back into this paper about why it's so unique. Okay, let's just go through it. So, this came from University of New Delhi, 2 prestigious universities, and all these people were involved. Class 1, top of their game. They're, setting the stage for innovations in India. You can guarantee it. All the smartest doctors are from India, right? Obviously, they know what they're at. So, right into it. We're going to check out why all these proteins matter and an uncanny similarity of unique inserts into this new virus. Spike protein, HIV, glycoprotein 120. So we found 4 insertions into the spike glycoprotein S, which are unique to the coronavirus and are not present at any other coronaviruses. Importantly, amino acid residues on all 4 inserts have identity or similarity to those in the HIV glycoprotein 120 and the amino acids. Interestingly, despite the inserts being discontinuous on the primary amino acid sequence, 3D modeling of the virus suggests that they converge to constitute a receptor binding site. That's a lot. So, yeah, these special little proteins and amino acid residues that they're talking about, they're on the place where the communication happens. They're enhancing this communication method, basically, is what's happening. Okay, so the finding of the 4 unique inserts, all of which have identity similarity to amino acid residues in key structural proteins of the HIV one, is unlikely to be fortuitous in nature, meaning it didn't happen by chance. This was designed. That's exactly what that means. So, a little bit about coronaviruses. They are single-stranded positive sense RNA viruses that infect animals and humans. There are 4 classes of coronaviruses. And that's just a whole lot of blah, 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 blah. So, to the next important piece. The spike glycoprotein, which is the protein glycoprotein 120, which is why, it's very unique. Well, that's why we're talking about it, this specific protein. The protein of the coronavirus is cleaved into 2 subunits, S1 and S2. S1 subunit helps in receptor binding, and S2 subunit facilitates in membrane fusion. So one does the handshake, and one pulls it in. Right? Those are the two units that they're talking about when this protein is cleaved, right? It's got two units that help the virus infect you. These subunits are the binding site receptors, and then they need co-receptors. So like it's a whole lot of science, but it's pointing a very big finger at man or someone, you know, who definitely had their paws all over this. So, on careful examination of these sequences alignment, we found that the spike glycoprotein contains 4 insertions. To further investigate if these inserts are present in any other coronavirus, we performed a multiple sequence alignment in the spike glycoprotein amino acid sequences of all available coronaviruses. So they basically, what they did is they took this coronavirus and they compared it to all the other ones that we know of in our database. And what did they find? We found that these 4 inserts, 1, 2, 3, and 4, are unique and are not present in any other coronavirus analyzed. Why is that important? Well, this coronavirus seemed to have veered off the evolutionary path and took a whole path of its own, and it just doesn't fit the bill. Right? So these 4 insertions, and they're calling them insertions, because they just don't align with anything that would be something that the virus would develop on its own. All right, so that would have had to got there. Right. So they went through the whole genealogy and tracked it back. The closest one they could find it to was the 2003 SARS. And yeah, so interestingly, all 4 of the insertions were absolutely 100% conserved in all the sequences analyzed. So meaning, okay, that these specific inserts, right, they're not changing. They're conserved right through all the strains that we know of. Like everybody that gets infected with this, they take a sample and they analyze it. They do the epidemiology on it, and they put the genome sequence in a database, and that's one. And then another person gets infected, and then they do a test on him, and then they do all that stuff, and that gets uploaded. Each strain of this, or each person that gets infected, like that they do a test on, it has a genome that gets uploaded, and they're comparing all these different genomes, and every different genome is carrying the same characteristics here. It's carrying this same insert. It's not varying, which means that it should. I guess it shouldn't. It's another sore thumb that sticks out. It's being conserved, considering the high variability of this reacting and interacting and changing. These specific proteins are staying the same, almost like they're purposefully being conserved to do a specific purpose. What would that purpose be? Communication between our healthy cells, our T cells, or whatever other cells it can infect. We don't know. Like, this thing is obviously gone to some length, so how far down the rabbit hole does this thing go? Right? Like, where does it stop? Obviously, these proteins are just tip of the iceberg, really. This is the one thing that we noticed on the surface. So what else is up with this, right? And the science is here. Everybody said blah 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 with this before and didn't want to believe it. But it's the way that this infects us is not the same. Like I said, it travels like the flu, but it is infecting us like HIV infects us. It's a lot. It's a lot. it infects us by using these CD4 cells. Alright? And where are CD4 cells? There are immune cells, our helper T cells. That's why they can't detect this. That's why it's showing up and it's disappearing, showing up and it's disappearing. The tests aren't catching it. You know what I mean? That's because this thing can hide from our immune system. It's not, our immune system's not producing any antibodies from this. There's no reaction happening. So they in turn can't actually look for the signal that's taking place to see if this is present and active. It has the same functionality or same characteristics as an HIV virus does, in the sense that it can go into like a state of remission or like a sleep. And they've confirmed this. I've heard reports that they've had virus particles in the samples, but they said that they're not active. But you know, the virus isn't doing anything. They don't know what that means. Well, I know exactly what that means. You know, the virus has gone into some stage of shutdown. I don't know whatever it's doing. Maybe it's mutating. You know, maybe it's, you know, we don't know much about this. We really don't. We're only going on what China's telling us. But there's videos coming out of China and it's batshit crazy. And you can guarantee it is. Because that city and those places shut down real quick. There's 1.4 billion people in China. And everything is shut down. The mail's shut down. Nothing's in or out. Economy's going down the trank and going like every day. It's just, it is. So what does this all mean? You know, it's an informational guideline so that you can take your own advice on it. And do some research. Do your own research and just be like, fuck, I don't know, Google coronaviruses and how they infect you. And then Google this specific virus. Actually, you know, you got to Google HIV-1 and Google the glycoprotein and the glycoprotein structure 120 and binding sites, but the glycoprotein is where it's all at, right? Because that's the communication center. That's what makes it all special. That's what makes it so different. And we know for sure that this isn't like anything we've ever seen before. And the science, there's another paper. There's another scientific paper right now. There's a second scientific paper. And this one here has been peer reviewed. Has been peer reviewed and has been accepted for publication. ResearchGate. A pneumonia outbreak associated with new coronavirus, probable bat origin. Don't let the title fool you. Okay. Bat origin, meaning that this came from an infected bat and somehow got out of the laboratory. Alright? It uses the ACE2 receptor as the entry receptor. Alright? This is, you know, it's some crazy ass something or other. Let's just read the story, the write up on it, real quick about this paper. So, coronavirus may have started in a Chinese lab where researchers were attacked by disease-ridden bats, scientists have claimed. We were just looking at this paper. New paper from the Beijing-sponsored South China University of Technology casts doubt on the government's theory that this deadly infection emerged at a seafood market in Wuhan. These two scholars said that the city's Center for Disease Control, just about 300 yards away from the market, hosted animal laboratories for research purposes, including 605 bats. Their paper, titled The Possible Origins of 2019 nCoV Coronavirus, claims bats once attacked a researcher and blood from the bat was on his skin. He said that quarantined himself for 28 days after bats also urinated on him. Wow. paper, which only gave maps as evidence of their theory, said that genome sequences from the patients with coronaviruses were between 89 and 96% identical to the coronavirus originally found in the horseshoe bat. Okay, this is going to make sense a little bit more here. Eventually. The end of the video. Maybe. So it read, the report, it is plausible that the virus leaked around and some of them contaminated the initial patients in this epidemic, through those solid proofs are needed in future studies. Okay, bats are broadly considered to be the original host of the new coronavirus. Chinese scientists have previously said it spread to humans via an intermediate host animal, possibly pangolins. No. Pangolin is like a armored shelled anteater. It's literally has like an armor shell, like an armadillo. And how's a bat gonna infect that thing? No, and it eats ants. That's all it eats. It's like, it's like I don't know. I guess it could happen, but whatever. Although evidence proving this theory was never published. So, they said that, but nothing ever came about. It was just a bubble to keep people guessing. As well as being close to the infamous seafood market where wildlife animals were being illegally traded, the lab is adjacent to Union Hospital, where the first group of doctors became infected. So, there's the seafood market. This is the Wuhan Center for Disease Control and Prevention, where they say these bats were housed, where they were doing research. And they were there. They were, in fact, doing research on bats there. And the hospital is just right down here. The hospital is over here. And, all in this vicinity, right? Because it went through that hospital like wildfire. So, adding weight to their theory, testimonies from 31 residents and 28 visitors to Wuhan suggested bats were never traded at the market or consumed locally. So nobody ever did that there. The scholars said that the closest native bats to Wuhan were around 600 miles away, and that the probability of them flying to the city was very minimal. Like, yeah, you gotta think, like, Wuhan's like a big metropolitan, it's a big city, it's a concrete jungle. Bats live in caves, not in the city. Anyway, the paper said the killer coronavirus probably originated from a laboratory in Wuhan, as well as the Wuhan Health Center for Disease Control. The report suggests that the Wuhan Institute of Virology could have also leaked the virus. And it's not very far away from there either. The report said this laboratory reported that the Chinese horseshoe bats were natural reservoirs for the severe acute respiratory syndrome coronavirus, which caused the SARS outbreak in 2003. And this specific SARS outbreak in 2003 that we're talking about is the same one that was that we were talking about earlier in the video, where he was doing the genome sequencing on it and saying that it is very close to this, but it's not quite this. SARS. This Wuhan virus is definitely in a class of its own, let's say. So, SARS is a member of the coronavirus family, and it killed over 800 people when it emerged in southern China 17 years ago. Emerged in southern China. The new coronavirus, officially known as COVID-19, is far deadlier, with the number of fatalities soaring past 1,500 this week. Well, we know those numbers aren't right. So, speculation about its origin. Experts have warned that fake news and conspiracy theories could lead to greater number of cases. Really though, right? You know what I mean? there was actually news reports where somebody said it was, I don't know, basically saying that, if the more false news you spread around, you're going to make the virus spread quicker. If you're spreading fucking, but regardless if it's fake news or not news or any kind of news or whatever you're spreading, it's not going to, anything on your social platforms or networking is not going to change how the virus is fucking moving through nature. But I did remember seeing that. Yes, actually, I think I watched this. So, anyways, back to this. Earlier this week, scientists at the forefront of the effort tried to track the deadly outbreak. Rubbish claims that it was genetically engineered in a Wuhan lab. One source of the rumor was a paper published by researchers in India, which was later withdrawn. Alright, and this is the one we're talking about. The paper in India came out of the Indian Institute of Technology. That's the paper. That's what they're saying. So, keep going, I think, maybe. Where was I? Damn it. Was I here? Here it was. So, Trevor Bradford, of the Fred Hutchinson Cancer Research Center in Seattle, said that an international conference, there's no evidence whatsoever of genetic engineering that we can find. The evidence we have is that the mutations in the virus are completely consistent with natural evolution. Imagine. Really. There's a lot of people that seem to think otherwise. And even here, Daily Mail's running with this. First coronavirus patient had no connection to the seafood market. So, did the disease start elsewhere? Huh? Sure seemed like it. Right? And people were just like, oh my fuck, there's people just dying everywhere. Like, just dropping on the streets. You see in the videos. Like, and it didn't come from, like, as twisted as the stuff they do there. Like, wow. But yeah, no, it's this is specific in its host specification, and the fact that it's being transmitted human to humans asymptomatically. It's attacking our T cells. It's hitting our immune system. It's doing things exactly exactly suspiciously. How's that for an answer? There's a fucking ton of stuff going. There is. And it's not just the science. There's science, there's questions, there's fucking speculation. Everybody's got something to say about it. But what we know from the World Health Organization is jack shit. But there's a lot of people talking with a lot of speculation that this thing is not natural. And I've been thinking that right from day one. As soon as Wuhan's right here. I don't know if you can see. I don't know. I guess you can't. Anyways, it's in the lower right hand corner, I guess, but it's central. I can't describe it. It's in there anyway, Wuhan. And they locked down the first city of 11 million people, and I thought then that was something to be like, what the fuck? This shit's crazy, right? And then it just kept going out. And then it infected everything. Like, everything is infected over there. They honestly, I'm gonna tell it straight how it is, and there's a lot of people infected by this, and it's putting people through a fuck of a time. I promise you, there's people out there and they're not having a good day right now. And they didn't do anything to deserve this. It hits anyone without prejudice. There is no majority, or age group or demographic that this selectively hits at. No. When they say, well, old people are more susceptible, well, yeah. Old people are also more susceptible to exercise. It's fucking common knowledge. It's not, they're not going to come out and tell us what this is. They're downplaying everything. And if you see it, they hang on these things that they can downplay. It's all over the media. But they downplay everything. There's nothing in there that they're trying to... It's all downplaying. Even when they're reporting on shady stuff, it's still being downplayed. Like it's not nothing. But we got 1.4 billion people cut off from the world. The markets are crashing. Apple lost fucking 5% on the first day of opening. $72 billion. Apple alone. But everything is shut down in there. Apple was just the first one to come out and publicly say, hey, we're going to take a loss from this. And Walmart, everybody was expecting Walmart to say something. They're still hanging on, but you know what? It's I think within the next however their business model works, their next meeting they're gonna have to announce it too. Right? There's companies shutting down everywhere that rely on items that come out of China. Medications. They already think that there's going to be a massive short of medication come the end of this month. 10 days. They figure, because whatever's out now, it's actually the supplies are dwindling because there's nothing more being made. All the generic medications, most of them are made in China. Like, most of them. I'm not exaggerating. And most of the car parts, most of the electronics, most of everything. You go look at something in your house and pick up the bottom of it and see where it's made. See where it says, and just go pick up 10 random objects in your house and see what it says on the bottom of where it's made, where it's from. It's not anything that they should be downplaying. They should be giving people a heads up. So why are they not? Why are they telling people, hey, this is... No. They can't do that. People will get scared and they'll stay home. Everything will start shutting down. But if you don't do that, if you don't keep people home, the virus just keeps spreading. You do keep everybody home, the whole economy and social fabric and society collapses. There's no other options. It's either we let it keep spreading person to person, or we cut our losses and we barricade right now. Maybe the far-reached cities or towns, out of the big metropolitan areas might still have, but you never know, right? You could have a super spreader in that town and just could be thousands infected, right? And all it takes is a couple of days and this thing can infect hundreds of people in a couple of days. Hundreds. If there's one person infected in a town or a city, a couple of days to be hundreds of people. Depending on that person's associations and if they're just stuck in their house all the time, well, of course, they're not going to be able to spread it. But let's say, the person's a social butterfly or whatever, out mingling and talking and, being infective and not knowing that they're infective. Just spreading like wildfire, right? Maybe that's why they're not telling us. Maybe they know it's already too far gone. So what do? You just don't tell anybody. In a situation like that, you wouldn't tell anybody. If it was already too far gone, you wouldn't tell them it was too far gone. Because then that's just... You just wouldn't do that anyway. The media is not going to come out and say... Or nobody's going to take a stand somewhere and say, well, you know, President of the United States isn't going to get up there and be like, well, guys, you know, we tried. Sorry. Oh, fuck. Lost this one. See you in the next round. Do you think they'll come out and say that? I truly believe, and I'm not trying to invoke fear or panic. And if you panic, that's only going to cause you to be more vulnerable, right? Fear is good because fear gives us a sense of heightened senses to protect ourselves, right? If we're not, you know, if we're just being nonchalant and just doing our thing, we're kind of sloppy, right? We just do things, whatever. We're not really paying attention to what we do. You know, fear gives us a sense of a heightened awareness, so we're more aware of the things we do, what we touch, what we see, where we go out, who we interact with. But don't panic. And there's going to have to be villages, obviously. Well, not villages, but I don't know how long this is going to take to burn out, or if it will burn out. Who knows where it's at? You know, even let's say, okay, we could take the assumption to say, okay, well, it did leak out of a lab. You know, oops, got out. But now that it's out in the population, what is it actually doing on the virology, on the virus side of things? You know, what as a virus is it doing? Okay, of course it's spreading and killing people, but it's changing. And you can guarantee it's going to only get stronger, and it has been getting stronger. In case you haven't noticed. When they came out and did the world big address to the world, they were like, well, there was only one case outside of China. You know, don't worry, it's all good. You know, one case, but it's just still, it's serious. It's serious, but don't worry, there's only one case or two cases or... And then there was like 20 cases. You know, it's still serious, don't worry, you know, it's only 20 cases. Don't worry. Don't worry, it's good. Now it's up to like, I don't know, 1,000, over 1,000 cases worldwide outside of China. Deaths starting to pop up outside China. Iran just reported 2 deaths from the coronavirus. Bam, bam. People are going to start dropping from this. And they can't hide it. It's just going to be just like China. What China did. They were like, well, fuck, what do we do? Do we shut down? Do we say go? Do we say stop? Well, we need more data. We need more data. And then by the time they even knew what was going on, it just... The hospitals were overflowing, people were dropping, dying in the streets, panic set out, and fucking chaos ensued across the fucking country. And now it's just rippling across the world. You gotta take the precautions that you think you need to do and keep yourself and your family necessary. Or safe, right? I have. Best as I can, right? And that's all anybody can do. But I think in the next couple of weeks we're going to see something. Maybe even sooner than that. Or maybe even longer, who knows? I'm not trying to be a pessimist or over skeptical, but it's just the nature of the facts. There's a lot of mystery shrouded in this. You know, why did China arrest the doctors and cover it up? People over there are fucking outraged at their government. Outraged. China just kicked out 3 Wall Street journalists, gave them 5 days to get out of the country. Gave some bullshit reason, like they, I don't know, but whatever. Something about racism and all this stuff. But regardless, China found a way to, you know, get these people out of the country, so now they gotta go. The reporters gotta go. The US reporters. Right? And this is in Beijing. Because Beijing's the capital of China. And they were able to hold the other cities down pretty quick. But Beijing's a little different. Right? Beijing's not as easy to hide. And on the world stage, Beijing would make headlines bigger than any other place. And especially on the global level of the economy, which China, this isn't a health crisis to China. On the highest levels, what they're dealing with is a political crisis. That's how they see this. They don't see this as a health crisis. They see this as trying to save face. And they're trying to dummy the numbers, and this thing is fucking vicious, I promise you. Whatever it is, regardless, it doesn't matter. It doesn't matter where it came from or how it got out or whatever the fucking thing is. Fact is it's coming. And it's coming like a fucking freight train that you can't see. And I'm not trying to be over dramatic about this. I'm trying to just tell it how it is. I'm not going to sugarcoat it. And I've been trying to save it for a long time in my videos, and it's hard to get my thought processes out in a sequential form where things fucking flow and make sense. Because there's so much information I'm absorbing and trying to process and trying to reiterate. Because I do see it, but it's hard to give information to people and just give them word of mouth. Like, well, just take my word for it. This is the way it is. I'm trying to back my words for it with fucking the best information that we have, given the current circumstances and how everything's evolving right now. We don't have time for all this stuff to get fucking published and go through medical journals and all this. This stuff's happening fast. It takes months and months to publish papers. It takes a lot of work. A lot of people got to go be involved with it, and it takes a lot of channels. It really does. It's not like, it's a process. So we go with what we got. And we gotta, use a little bit of discretion, of course. Certain things. But like, what the fuck? You can go to jail in China criticizing Beijing's coronavirus response. Do you believe it? I do. And not only do I believe it, that's the most vanilla way in the Western media that we could put it right now, right? They're killing people over there. Flat out making examples of people, because you know why they're killing people? And they have no fucking problem doing it. And you know why they have no problem doing it? These military and combat people going in and taking control of these cities, they were raised in a communist country, China, right? And they were selected. And he recruits these people from like young ages and brainwashes them. These people, or soldiers, or whatever, dogs, whatever, they're conditioned. They are literally brainwashed and conditioned to be a certain way and have no heart and just react. They follow orders to the T. They really do. They're like robot soldiers. And they have no compassion. They have no fear. They're just ruthless. And they're setting examples so that people will run indoors. They want to scare people. They want people to see what's going on. So everybody hides. That's what they want. Everybody indoors. If you're caught outdoors, especially caught reporting or doing anything along the lines, let's say, criticizing the government's response, like throw you in jail. And propaganda, yeah, oh my god, it's disgusting. All you gotta do is go, if you got a Twitter account or wherever, Google, just go search China Daily or CCTV. It's the two state media platforms there, and everything's just rainbows and fucking butterflies. Oh, it's disgusting. They're still not admitting to their citizens that there's even like, it's like they're like, well, you know, our quarantine measures are really showing some positive effects, and we're really getting a handle on this, and our workforce is getting stronger, and we're, you know, it's, yes, it's nice to fucking, you know, I guess have good intentions and good dreams, or, you know, but let's save the dreaming for after you do some actual action, because, you know, there's nothing actually happening. It's except the virus getting stronger and tearing that place apart. But they're hiding it at every corner, like it's not happening. And they even told them, our government told them that they have to actually now, the media outlets there, yeah, they have to operate at the same, like they have to be honest with their citizens, basically. Um, there it is. U.S. to treat Chinese state media like an arm of Beijing's government. You know, the media there is flat out lying to them. Like, and everybody knows the difference. Of course they do. Like, they can see what's going on with their own eyes. You know, they can. So, Washington CNN. United States will begin treating the 5 major Chinese media companies as extensions of Beijing's government, requiring them to comply with rules governing foreign embassies and consulates. So, the propaganda machine. They're just spreading bullshit. Like, why? Sam, yeah. Uh, I'll see if I can fix that so you can send some links. Let me see what I can do for your dog. I don't know if I can do. Maybe. I don't know. What's this do? I don't know. I knew there is something somewhere that you can like send the links, but I don't think I can. I think I can't do it while it's streaming. But yeah. The peer review paper. Are you talking about? You're talking about the second one that came out of South China. This one here. This is a peer review PDF file. I don't know if that's the same one you're talking about. Ammonia outbreak associated with coronavirus. This one was written. It was written, or it was received by this company or this platform January 20th. It was received by ResearchGate January 20th. It was accepted January 29th. Accelerated article preview published on February 3rd. Yeah. But then, yeah, there's this one, but that's not peer reviewed. But back earlier in the video, go through the structural protein.
Hi.
And he makes some similarities. He makes some Yeah, he makes some crazy similarities. Oh, your paper 2004? Oh, okay. No, I'm not sure. Um, I do have I do have another report from 1999 about a coronavirus that they made in a laboratory setting. It wasn't the same kind of thing, but um, yeah. But there's like a lot of wow. There's a lot of skeptical things floating around, right? And it's not so much a conspiracy as it is like a certainty now. Just given the way things are, it really is. Um, and we all know that China was involved bioweapons research. Like this is from Wikipedia. China and weapons of mass destruction. You know they had a program going since the 1980s. Fucking biological weapons program since the 1980s. And of course, yes, they signed the Biological Weapons Compact, but did they actually stop producing it? Did you? I don't. I don't think so. You know. And then we have a defector here from the Soviet warfare program, and this is him here. And there's a book that he wrote. Defector tells Soviet Union, and this was on the New York Times again. Um, and they confirm it, right? And he even says something crazy here. He says the book also reports that Soviet researchers tried to turn HIV into a weapon. Soviets tried to do that, not the Chinese, but the Soviets. But there was a very big population back then, and when everything got broken up by the compact and the inspectors went in and everybody went in, all these people, um, there was about 40 institutions that were part of this group, um, and they were used exclusively for offensive agents and the weapons of military. He wrote. So these were specific bio labs, right? And top secret program obtained a sample of HIV from the United States in 1985 and tried unsuccessfully to turn the slow killer into a weapon. And this is William C. Patrick, a key figure in the United States former germ warfare program who helped debrief Albeck after his defection in 1992, said many of the book's assertions were consistent with what he told the U.S. officials at the time. Wow. You know what I mean? But that just, that's. It's a lot. Yes, it's a lot. Is it stretching? I don't think it's stretching too far. I really don't. Like, China's had a fucking biological weapons program since the 1980s. Of course they're gonna have knowledge, right? Like, they are setting the stage. Actually, you know what? Let's look at a video from the Wuhan Institute of Virology of one of their researchers going out and collecting bat specimens. Yeah. It might take me a second to get this up and rolling the way it should be. Bear with me. Yeah. And I think I have it in another spot. Oh my goodness. Okay. Yeah, we gotta find that video. Hmm. Um. Yeah. My little fellow watches a lot of stuff on here too. So, um, here it is.
Can glasses improve my hearing? What's PD? How do I buy online? What's blue light?
Okay, and if you see right down here, you'll see SMG is funded in part or whole by the Chinese government. Yeah. So, just skip this. The Wuhan Institute of Virology. Let's just check out. I'm gonna go for a quick cigarette. You guys watch this. Oh, Sam, yeah, man, sorry. I'm so bad at they're trying to make SARS vaccines ages ago. They failed miserably. Dude, yeah, you know what? I know. I understand the vaccines for the SARS. They failed. Yeah, this one here is unfortunately it's not even it's a close relative to SARS, but it's extremely different, and I'm still trying to figure out the best way to process that to explain it to people without sounding like a complete dummy. But here, let's just look at these researchers from the Wuhan Institute of Virology. This center or a place where the Wuhan Institute of Virology. That's the seafood market. This isn't the virology one, but let's just say it's very close because it works out the same way to the. But this one here is only 300 meters basically, but the Institute of Virology, the Wuhan Institute of Virology, is only 20 miles. Actually, I think it's even less than that. It's like 17 miles from the seafood market. So, without further ado, let's see what they do when they're collecting their bats for the Institute of Virology. And this is the actual scientist. He's the man. Probably created this, right? Let's just look. They're going out to get bats from their natural habitat. Way, way, way out of the city, remember?
声音被呼吸吞没，身体和黑暗融合。当我们面对一个看不见的敌人，只有隐藏自己，才可以发现隐藏更深的威胁。我不是医生，却在做治病救人的工作。我不是战士，却守护着一条看不见的国防线。
至于它里面的结果是生理正中间这里，按这个来说应该是成。
我是田俊华，从事病毒样本采集和分类工作，是一个隐形边防线的守卫者。从古至今，病毒就是人类生命的杀手，极强的隐蔽性，让人猝不及防。病毒肉眼不可见，可以通过病媒生物传染人类，而病媒生物在进化过程中与病毒共同依存，可能形成了抑制病毒的某种物质。我们只有发现更多的病媒生物，才能为这种疫苗奠定重要的基础，而这正是我们肩负的使命。跟踪、潜伏、 观察。山野是我的办公室，山洞是我的工作台，
这个吃到一个熊的哇，太幸运了啊！
林地是我的实验室。湖北地区有丰富的病毒媒介样本资源，
能够把人咬死的这种皮，危害非常大。真抓了一个，
但一部分病媒生物生活的环境对人类来说非常恶劣，是一个极大考验。在已知的生物中，蝙蝠体内病毒较为丰富，可以找到绝大多数的人类致病病毒，狂犬病毒、SARS、埃博拉病毒。就此，蝙蝠栖息的洞穴成为我们的主战场。潮湿、黑暗、孤独，蝙蝠的生活环境都在人迹罕至的洞穴里。但只有这样的环境，才能找到最理想的病毒媒介样本。
最安全了。这里的蝙蝠它主要是菊头蝠和蝠翼蝠。
如果说我们的皮肤裸露，很容易接触到蝙蝠的排泄污染的物体，所以说这个风险比较大。要探寻出一个蝙蝠族群和病毒的共生关系，往往在山洞里要住上几天。没有手机信号，没有补给，是真的害怕。怕感染，怕迷路。因为害怕，所以我每一步都很小心。越是害怕，就把每一个细节做好。因为你发现病毒的过程，也是最容易被病毒感染的过程。这是一场没有硝烟的战斗。
你得把它拉开，就是这样。一定要把这个洞封死。出来一个，哺乳动物里面唯一能够飞翔的一个动物。要不然它把网抓破了以后，我们来做采集标本，然后做它携带的一个病毒的研究。好，发现一个蜱虫，这是长腹硬蜱，非常特殊的一种蜱，别的地方抓不到的哇！今天真是收获非常大。
这是一条未知的路，就像蝙蝠生存的洞穴，深不见底。你望着深渊，但是并不知道是什么也在望着自己。
这东西是别人不好替代的。
身在其中，
做任何事情，你只要自己。
我学会了对自己的审视，克服了恐惧。
有时候需要家里的人来支持，习惯了，挺有意思的。
路虽然不好走，但必须学会在迷茫中坚持自己的方向。在过去的10多年里，我们走遍了湖北的每一个角落，探索了几十个没有开发的洞穴，研究病毒媒介300多种。但我却希望这些病毒样本只是作为科研而留存，永远都不会有应用的一天。因为人类需要的不仅是疫苗，更是一份来自大自然的守护。
I'll pause that right there. You see, nearly 2,000 types of viruses have been discovered by Chinese CDC authorities over the past 12 years. Only 2,284 types of viruses had been discovered worldwide over the past 200 years prior to China's discovery. China has taken the lead in the world in the field of basic research of virus. Interesting. Just saying. It's interesting. Let's just see what this guy has to say. The ambassador. Alright? I'm gonna start getting really cranky here. Cause it just stinks. It does. It fucking reeks of cover up, of mismanagement and miscommunication, just dropping the ball. Really. But anyways, here. Let's I'm just gonna keep painting a picture for you. You know, maybe you'll come to the dark side. No, it is what it is though, right? It is what it is, and let's hope it's not. But I've goddamn prepared. Like it is. I prepared for the worst. Sam, what are you saying, dog? Oh, it's also been like a feel from the beginning that this is no natural animal. Yeah, I didn't understand the whole science from the beginning, but I just knew from the magnitude of reaction. You know what I mean? Like it was like, what the fuck? Actually, you know what? Let's just go look at a quick timeline of events. Let's just see. Cause yeah, Wiki has done a great job of putting this in a chronological order. My Facebook wall is also a chronological order. I must have posted at least 20 things a day since this started. Like, I'm not even kidding. When I go to look for something, it's just like, oh my fuck, it's gotta load forever. But anyway. So, this is kind of when it all started. Outbreak chronology. So, on December 31st, 2019, a consortium of Chinese medical experts were charged by the Chinese CDC with investigating the inception of what was commonly known as the Wuhan coronavirus. On January 24th, 2020, their report was published in The Lancet. They noticed from their review of local medical records that the person later to be diagnosed with the Wuhan coronavirus had first presented symptoms on December 8th. However, consortium found an earlier case of a person who had experienced symptoms on December 1st, pointing to an even earlier origin. Apart from this early case, between 8th and 18th of December, 7 cases later diagnosed with Wuhan coronavirus were documented. 2 of them were linked to the seafood market, 5 of them were not. They said it came from the seafood market. But there was 5 people who had no connection to the seafood market. Never went there. So, on December 12th, Chinese state broadcaster CCTV reported in a broadcast airing on January 12th that the new viral outbreak was first detected in the city of Wuhan on December 12th. That's when they said it was first detected. CCTV. But medical records state at first it was December 8th, but then later on they were saying that there was a person who had experienced symptoms on December 1st, pointing to an even earlier origin. So it's big. Chinese epidemiologists with the Chinese Center for Disease Control published an article January 1st stating that the first cluster of patients with pneumonia with unknown cause had been identified on 21st of December. So it's just, you know, you got the CCTV reporting, well, January 12th. You got CDC saying December 21st. And you got medical records pointing back to December 1st. Mm-hmm. Mm-hmm. It's a wow, a lot happened. Let's see. January 21st. Aha. A total of 291 cases have now been reported. 291 January 21st. And what's today? February 21st? Even the reported numbers, which we know aren't right, seriously, they're just not. They can't be. The testing capacity, the capabilities, the logistical nightmare that's happening right now. And the quarantine measures, like, nobody's going to the hospital. Anybody who has a fever is not dare going to the hospital right now. Could you imagine? What they're doing is they're just taking you and putting you right in quarantine. And if you have a fever, they can't test you. So what they do is they quarantine your whole family. And if you're in an apartment complex, they will fucking board up the whole apartment complex for 2 weeks. Nobody in, nobody out. Doesn't matter, fire, nothing. They're welding the doors shut. You think I'm crazy? I wish I was. This is the extreme lengths that these people are going through. And it's fucking over the top, beyond, like, and these people didn't deserve that. You know, it's not like, you know, these people didn't deserve to get infected. The shit that was going on, it's like, look, this here, fuck. Oh my god. Oh yeah, we were gonna look at that video. I get rambling, but look at this shit. This is what they're doing, and you know why it's real? Like, everybody's all in hazmat suits. I've seen this video a few times. These people, for whatever reason, either they're from a different city or whatever, but they're getting quarantined, and they're going in the back of this fucking box. Right? Wow. Let's just see how they're responding. And these are like, they look like university people or like something, right? Like they're young. And like they're hugging and like she's crying, and I guess they're telling her that like they gotta go, right?
抓起来了。
哦，进去了，看到没有？
他们。
哇，太可怕了。
我不喜欢抓进去。
可是他们生病了。
所以他们要去哪里？
Right? Tell me that it's not happening. China slams conspiracy theories. Alright, let's see what China has to say about the conspiracy theories.
Because it also gets at, there's a lot of unknown and a lot of suspicion because of that. And in fact, this week, Senator Tom Cotton, who sits on the Senate Intelligence and Armed Services Committee, suggested that the virus may have come from China's biological warfare program. That's an extraordinary charge. How do you respond to that?
I think it's true that a lot is still unknown. And our scientists, Chinese scientists, American scientists, scientists of other countries are doing their best to learn more about the virus. But it's very harmful, it's very dangerous to stir up suspicion, rumors, and spread them among the people. For one thing, this will create panic. Another thing is that it will fan up racial discrimination, xenophobia, all these things that will really harm our joint efforts to combat the virus. Of course, there are all kinds of speculations and rumors. There are people who are saying that these viruses are coming from some military lab, not of China, maybe in the United States. How can we believe all these crazy things?
You think it's crazy. Where did the virus come from?
Absolutely crazy.
Where did the virus come from?
We still don't know yet. It's probably, according to some initial outcome of the research, probably coming from some animals. But we have to discover more about it.
Oh my fuck. Did you hear what he said, that the virus was coming from the United States? All these crazy stories that the virus is coming from a lab in the United States.
You think it's crazy. Where did...
And rumors. There are people who are saying that these viruses are coming from some military lab, not of China, maybe in the United States.
Oh. Oh my god. Right? Fuck, it's like such a cover-up everywhere. This guy's like pointing at it in a scientific way. You know, this is back in the first of the video. If you want to go back and check out the first of the video. Yeah, it's just, it stinks. It stinks. Look at this. When was this? Yeah, remember? January 29th? This was a while ago. And this was when he ordered the military. But you know what I mean? Like, you could... Oh my fuck, those poor fucking people, man. Like, they didn't even have a chance. Like, their country lied, like their government lied to them. Flat out told them like nothing's wrong. Still saying that there's like nothing fucking happening. Like, oh really? It's great. Sunny days, another day in paradise. Like, fuck. Unreal. I don't get it. Actually, you know what? Let's go look at China's. Let's go look at their media. China Daily. This is their, you know, this is the state-run media. Let's see. Experience in fighting virus can be shared with the world. Technology. Yeah, look, everybody's doing great. He's got a camera up there pointing at something, and there's some people doing some stuff there. Opinion, video, business, culture, travel, China Watch. What's this? Nothing about the virus. China's positions on major global issues. Chinese government public positions on some major global issues in the 70s. Yeah, so everybody's... China brings benefits globally. Rise of China. Tariff cutoff on auto imports to benefit buyers. You know, everything's, you know, they got some numbers over here. Everything's, you know, this guy's got a pencil. He's doing all right. But you know what I mean? There's nothing in here in the virus. This is China. This is, I don't know. Actually, do you know what? We can get it better. Let's go to Japan Times. Because there's some stuff going on down there. Their media is kind of, you know, still got a little say. It's part of China, but, you know, Japan's kind of a little more democratic, I guess. So, SDF on Diamond Princess get more protection after chaos claim in video. SDF. Okay. It's an F. What the fuck is an SDF? Self-defense forces. Ah. I thought it had something to do with like up fucking shit creek without a paddle forces. Because really, like, what the hell were they thinking on this cruise ship? Like, even I was sitting back here going, what the fuck? I would even, if I was calling the shots, I wouldn't even do something that stupid. Like, fuck. You would at least take the healthiest ones off and leave the sick ones on the ship, I would think. Because you don't want to get this thing in the fucking hospitals. Right? It already infected multiple hospitals and the staff and killed numerous people, like, at the hospitals, including the one most notably the director of the Wuhan hospital. Killed the director. He died of coronavirus. So why would you want to take the risk of bringing this into your hospitals where you already have patients, where you have people possibly, you know, delivering babies or whatever the fuck's going on? You know, I don't care if you're using vacuum sealed tubes or whatever the fuck you're using. This thing is extremely contagious and transmissible. The science shows it. And it's like, what the fuck? What are they thinking? Like, honestly, if I was running that shit, I'd be like, okay, the sick people stay on there. We'll bring the hospital to you. Don't come on land. We'll get the healthiest people off, and we'll still quarantine them on land, but we'll quarantine them in groups instead of like one great big fucking group. And you would level the groups out, of course, by the most, as best as you could, from most possible contaminated to the least possible contaminated based on fucking whatever variable factors, right? But oh my fuck. I just shook my head at that. I couldn't believe it. And like, they were just sitting ducks. If you weren't infected before, the longer you just sat there in this fucking petri dish festering, and like, this thing was just circulating. Oh my god. Oh. And we don't understand the transmissibility of it yet. We don't understand exactly the methods of infectability. We don't understand so much. Like, we could have this literally. I'm gonna pull this out and just say, based on like a educated guess, that, well, they're saying now there is actually studies that there is like a 24-day period before you can show symptoms. I'm gonna say it's whatever the hell certain biological processes are present for you to express these symptoms or traits or whatever the virus is. It depends upon your biological processes more than anything, I think. And everybody's getting affected a little bit differently. It's like, it's very unstable. It's I don't know. It doesn't seem. It's just too much. It is. It's too much, and there's. You would never, unless you were like an absolute fucking genius. You know what I mean? So you got to be able to, like, at least be able to pull it out somehow and make sense of it somehow. And I'm trying to make sense of it in the best possible way to people about these proteins. And that's what this whole demonstration here is about. It's about these proteins. The glycoproteins, which they mentioned in the reports. That's what's so specific. That's what's so unique about this coronavirus. It really is. It's so unique because of these little proteins, and they aid in the communication between the virus and our healthy cells. The better communication they can have, well, the better interactions. And if it can interact with it, the cell will not recognize it as a threat, and it will allow it to basically infect our cell, right? It won't. This thing is infecting our T cells because it's infecting the CD4 receptor cells. And they're only, these CD4 cells are only on certain cells. They only, meaning like it's like a lock and key mechanism, right? You can only put the key in the lock that it's gonna fit, right? It's the same thing as these CD4 cells expressing this lock, and these glycoprotein spikes, they are the key, right? And when they get inserted and they fit, CD4, it's a glycoprotein found on the surface of immune cells like T helper cells, and also monocytes, macrophages, and dendritic cells. Like, those are the other cells, but the most important ones here that we can take away is it's infecting our immunity T cells. And why does that matter so much? Well, because our T cells are not going to be recognized as a threat by our body. So it's going to be very hard to combat this thing. It's almost like it was made to be extra vicious. I don't know. Science sure fucking points a lot of fingers. Let's go back. Let's just watch this again. This is the same. I'll run this again because we can't watch this as many times as we need to. And there's new viewers and stuff now, and I appreciate everybody tuning in. Meaning it's not simple to make a leap from infecting one kind of animal to the human ACE2 receptor. No, it's not. It's really not. For cross-species transmission to happen, Ethica 5, yeah, man, like, it's a big, the probability is exponential to have an intermediate host being able to transmit a virus that is already, let's say, adapted to that intermediate species host, and then makes the jump to us to amplify it, right? Something would have to take place in that process there. And there have been amplifying events. There have been. But this is the way that this structure is, specifically for this type of coronavirus, is unlike any other type of coronavirus except for the SARS 2003 virus. That's the only one that it's close enough to. But what's big, the biggest difference is that this one here has a couple of extra tweaks to it from the last coronavirus that a lot of people assumed somehow got out of containment in the laboratory. But, you know, that was 2003. This is 2020. Technology has come leaps and bounds. CRISPR wasn't even, I think CRISPR might have been just a bubble thought at this point in time, and then CRISPR is like a gene editing software that allows us to do this kind of splicing, which it's our technology has advanced a lot, so we're able to do things. And China has, you know, it's documented they have had a weapons program. You know, China's had a biological weapons program since the 80s. Since the 1980s, they were reported. Okay, but now it actually goes back. It actually goes back way back. I think to the 70s, in the Soviet Cold War era, like around something like that. But anyway, they have a huge history with this stuff, right? So they have an understanding of this stuff. They've been researching it for a while. And as the video was earlier in this show about the Wuhan Institute of, or sorry, the Wuhan Infectious Disease Control Prevention Center, showing how they caught the bats and going after these horseshoe bats that are natural reservoirs for the SARS virus. So anyways, there's a lot of mystery. Let's go back and we're going to go and check out this 3D expanded view of coronavirus under electron microscope. and then it's going to roll right into basically it's a little 10-minute video, basically giving an expanded view, comparison, contrasting certain viruses, especially the keynote, the HIV virus that he does make note in here for all the viruses as this is all the virus that he could have chose from to make an example of, but he chose the HIV virus specifically in this video, documenting and contrasting the coronavirus and the current novel coronavirus, or COVID-19, or SARS-2, or whatever you want to call it. But here it is. Glycoprotein structure and why it's so important. This is the communication network that allows the virus to actually infect our cell. Without this, it's like a car without a battery. It's a vital piece. You have to have it for it to function, kind of thing, right? But here's the communication center that shakes hands with our cell and acts like a Trojan horse and deceives our immune system and sneakily slides in, and before we even know it, we're infected. Before our cells even know it. And this protein is specific to this virus alone. And that's what we have to take away from this. The whole science behind it, I'm still trying to figure it out. Right? And I'm trying to lay it out as best as I can as I find it, because time is of the essence. And here we go.
So here's another structure, again of SARS, because we don't have the equivalent one for Wuhan. It's the spike, and instead of the red, blue, green coloring, I made it all white because I wanted you to focus on this colored part at the top. Okay, the orange is the differences, all the locations where there are differences between the Wuhan virus and SARS. And at the top, these colored blobs are antibodies. These are things your immune system produces. These are fragment antibodies, like the light green and olive. This is a light chain and heavy chain. An actual antibody in your body is about twice as big as this, but this is a fragment that they use in experiments because it's simpler to work with. So there are 3 antibodies, 3 pairs here. I've colored them differently, again, just to see, to easily distinguish them, but they're the same antibody. It's an antibody called S230. It came from a patient who survived SARS. And you see that it binds at the tip of the spike, right near where the ACE2 would bind. And this is a neutralizing antibody. And by covering the spikes of the SARS virus with the antibody, the ACE2 isn't able to bind, and then the virus isn't able to recognize cells and infect them. Okay, so this is a mechanism your immune system uses to fight off the virus. If it can't get into your cells, then it can't reproduce. All right. One other thing that I told you I was going to come back to was these sugars. And I've colored them pink in this structure, and they're all over the surface. And these sugars are a technique viruses often use to defend themselves against the immune system. Your cells in your body and proteins, molecules on the surface of your cells, like the ACE2, often have sugars coating them. And so your immune system does not generate an attack against sugars generally, because then it would end up attacking your own body. So the spike here has disguised itself with all these sugars. But the problem is, up where it needs to recognize the host cell, it can't put sugars there because they would be in the way. They would prevent the recognition with ACE2. And so that's a vulnerable part. And so that's where we see this antibody bound. Okay. When I showed you earlier that the tips of the spikes, the 3 copies were folded in, that may be a mechanism to disguise the virus from these antibodies and from the immune system. It keeps them folded in, hidden from view, and only occasionally flips them out to see if there's an ACE2 molecule nearby. All right. Let's see. I think that covers everything I wanted to tell you.
This was a rogue operation, that it was not authorized.
We still don't know what happened to the remains of Mr. Khashoggi. How is that possible?
We don't have a history of murdering our citizens.
16 months have
My
gone by
bad.
since the Saudi
I
journalist
missed that.
Jamal Khashoggi was killed
terrible.
by agents of his own state.
I was
in
making
Istanbul.
a coffee.
But the issue doesn't go
but I
away.
was watching.
My guest
Anyways,
this week here
that
at the
ended way too soon.
Identical,
What the
which
hell
is a
happened?
high image of these black arrows show two of the Wuhan virus.
Okay. Here we go. Sorry about
ongoing epidemic in China and is spreading around the world right now. Let's take a look at what a Wuhan
again,
coronavirus
Thanks
looks
for
like
sticking
in
around.
electron microscopy.
Watching that crap.
Okay.
And this crap. Again, the glycoprotein. Check it out.
All right. So I have an image of these black arrows show two of the Wuhan virus, two copies of the Wuhan virus. And you'll see that it's a round particle. And on the outside, it has these spike molecules. There are these little fuzzy guys around the perimeter. On the inside, there's RNA, which encode the instructions of the virus. And the virus's goal is to get inside your human cells, cells in your lung, to dump its RNA into your cells so that your cells can produce more copies of the virus. This strategy is not specific to coronaviruses. A lot of familiar viruses use these molecules that form spikes on the surface to invade human cells. Let's take a look at a couple other examples of that. This is an influenza virus example, and in the center is an influenza virus particle, and you see again the spikes, the little fuzziness along the outside. The two other things are just empty membranes. They're there to let researchers see how does the virus actually fuse with these membranes. So the idea is how does the virus fuse with the actual cell, but this is a simpler model of that. And we can see I can kind of slice through at different depths and at different angles. And if I get the right position, I can see where the virus is contacting this nearby membrane. It's formed this little cusp. It's going to fuse with it and create a hole so that the viral RNA, the genome, can be transferred into a cell. Let's look at one more virus that uses the same strategy of spike molecules on the surface. We'll look at HIV virus. So here are a half dozen HIV virus particles. One thing you'll notice difference, we see the membrane around the virus. It's spherical. Again, I'm cutting through it, and I can sort of slice in and out through the virus. And one thing we'll notice is that there are many fewer spikes. That influenza virus and the coronavirus were covered with the spikes. HIV virus is a little bit different. It only has tens of spikes. For instance, here's one, this little stub sticking out. Okay, so we're going to look at these spike molecules. They're used by the virus to recognize your cells and to enter your human cells in your lungs, the coronaviruses. In your lungs. We're going to look specifically at the spikes on coronaviruses. Okay, so here I've brought up some electron microscopy of human coronavirus, and it's looking kind of pale. Let me try hiding this and showing it one more time. Let me try to make the threshold. Okay, I just changed the threshold of this electron microscopy, the displayed threshold. So this is what one of these spikes looks like on a coronavirus. Okay, it's a human coronavirus. These human coronaviruses, there are 4 different strains that are known. They cause colds. It's a mild illness. They cause about 10% of the common cold, cases of the common cold. The virus would be here at the bottom, and this is the spike sticking up. So this is the top here is the furthest part away from the center of the virus. And this is what the electron microscopy looks. This more detailed view than what we were looking at a second ago was obtained by averaging images from like 10,000 different spike molecules to get a higher level of detail. And from this electron microscopy, we can figure out exactly what the molecular structure of the spike is. So let me show you that molecular structure. Okay, so here's what the spike looks like. And we have 3 proteins here. There's a green one, a blue one, and a red one. And they're actually all the same. They're 3 copies of the same one, and I colored them differently just so that we can clearly see the 3. The spike is symmetrical. Okay, all 3 copies are more or less identical in their shape. The orange part I'm going to talk about a little bit later are sugars covering the spike, sugar molecules. And the really interesting part of this structure is the molecule that the virus uses to get into the cell is actually here. And it's this little tiny thing. It's a sialic acid molecule. And it fits in this little cavity in the spike. The little blue dashed lines, let's see if I give you a better bet. There's some little blue dashed lines. Those are hydrogen bonds. Those are attractive interactions between the sialic acid and the spike. Okay. When the virus encounters one of your cells, and your lung cells and other cells in your body are covered with these sialic acid molecules, it knows it's found your cell because the sialic acid molecule sticks here and triggers the initiation of this reaction, which is going to make the virus invade the cell. All right. So that's a human coronavirus that causes colds, not the Wuhan strain. Let's take a little closer look first at this sialic acid. It's a very small molecule. I just want to briefly show you that. Okay, it's this little speck here, only a few tens of atoms. It's a sugar, it's a monosaccharide. And you might think, and I guess another important fact, influenza virus invades your cells and recognizes your cells by the same molecule, sialic acid molecule. Okay, so that's interesting. You might think that other coronaviruses, well, they're in the same family as this human coronavirus, would also use sialic acid, but surprisingly, that's not the case.
And now what he's referring to there, surprisingly, that's not the case. What he's going to go into here, the next one, is the Wuhan coronavirus, which is... And everybody's been saying it, and the research paper said it from the first, and everybody shot that paper right down, and, you know, I kind of kept my faith with it. It looked legitimate enough, and it looked right to key points. But yeah, even this guy here is just a third source now. You know, that's the scientific-based source that's confirming, 3 sources confirming that this isn't something that was, you know, necessarily evolved along the evolutionary lines, right? It didn't pick up these proteins magically by itself. It's 2 separate viruses, and he mentioned the HIV virus in here for a specific reason. And when he mentioned the proteins, or not the proteins, but the spike envelopes, or the protein envelopes, like the, you know, the spikes, what we're talking about here, he noted that the HIV only had a small couple of ones. Like, you know, he only pointed to one little spike coming off there, right? But we know the coronavirus is covered in spikes all around it. And the HIV virus itself only has one of these little tiny little spikes, or a couple of these little tiny little spikes. But this protein is so enhanced and so infective, it doesn't need a whole bunch of spikes, you know? It only needs a little bit, because once it gets you, it's like, it will infect you. And that's what's special about this glycoprotein 120. Alright? It's very unique to HIV of itself. And somehow it just found its way onto SARS, that came out of China, and now this. But this one is a little different. This one's upgraded. This one's like SARS, but it's you know, it's upgraded. That's just the only way to say it. But he refers to the HIV virus because it's using this mechanism, right? And he's a scientific person. He can't be biased to the situation, and he can't come out and say blah 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 blah, because this is coming from, obviously a place that has some goddamn good technology, right? So he's not going to come out and say this or that, but he's going to lay out the science. And if you pay very close attention about the cues and the things that he doubles back on, like he'll double back on things and be like, well, now let's go, I want to talk about this again, meaning there's an important there's an important thing here, so let's listen. All right, that's how he's communicating. All right, and you just got to be able to pick out the little things that he's saying. All right, and he's saying that this isn't right, and everybody's saying, all the scientists are saying this isn't right, so it's not right, meaning, god damn it, it had to come out of the lab, it had to have, and all the secrecy and the media with China and the fucking cover-ups and the rest of the doctors, and it just all fits. Right? It's just like a big nightmare story unfolding. But, it's about getting the information out there to people that can take this and use it to their benefit. Right? And nothing's concrete or written in stone here. It's not proven 100%. But if we were to look on an evolutionary curve as the way things naturally flow in a natural order, this is stepping outside the box. And, it just doesn't fit the natural order. And everybody's saying it. And I think we can all take, regardless, even if it, let's just say evolution or nature just did take a big vector off to the side, right? Like, it could happen. It can. Nature does weird things sometimes, but I don't think to the degree of this, right? But it's regardless, it's a lot worse than what we think it is. And it has capabilities of hiding, in a sense, from our own immune system, because it infects our T cells using the CD4 receptor cells, which is specific to this protein. That's how HIV infects us. It attacks our immune system. Human immunodeficiency virus. Immune deficiency. It infects us that way. But you can think of this virus, this Wuhan virus, this one that's going around now, essentially it spreads like the flu, but it's much more vicious and it's much more contagious, but it spreads in the same manner. Viral droplets, touching close contacts. This here is even seeing that it's now aerosol, so that it is in fact basically airborne. So that it spreads in that manner, but it will infect our cells like HIV infects our cells. That's what it's saying. This virus spreads like the flu, but infects us like HIV. And that's not saying that it's HIV. It's how it infects us. It's how the HIV is so infectious, because it can recognize cells and communicate with them very good. And what it does is it tricks the cell. It tricks our healthy cells into thinking that there's no danger. And it allows a process where it fuses with our cells, and then it infects our cell with our cell not even knowing what happened or what's going on, just totally oblivious to the situation until it's too late, and then, it's over infected. And that's what's happening. It's infecting us like the human immunodeficiency virus, but it's not that virus per se. It's just using that method of entry, which is very specific, unlike any other coronavirus, except the SARS virus. But this here is a little different than the SARS virus, and he goes into this, and the science behind it. I'm just trying to figure it out too, right? First, I was just drawn to the news about the news side of things, but then I was like, what the hell? You know, these scientific reports came out, and I googled every word in there I didn't understand. I just had to, because like it's that big. I feel like it's just fucking up, and it's information for people. Because what if the government doesn't give us a heads up, right? At least if you have an awareness about you might have that extra fucking minute or two that you could, you know, we don't know how this is going to happen or react. We don't. But we can just take a lesson from China, because we know that place went up real quick. Real quick. Military guards, checkpoints, barricades, city quarantines everywhere. It's just overnight. Just rolled right out across the country. I don't know. We don't know how bad this is. We don't know how infectious it is to a sense of what the latency period is. We don't know any of this. There's almost nothing we know about this. And the World Health Organization, from just, you know, the most recent one as of this video, as of the 21st February, they still don't know. And it was a very vague, broad, you know, even worse, one of the worst broadcasts they ever did, but the audio and sound were finally good, but they weren't talking about anything of interest, so of course the audio and the sound would be good, because we all know the audio and sound are shit through all those broadcasts for some reason. Like I just learned how to broadcast myself like within like 2 weeks or 3 weeks since doing this, and like I don't know, they need a new sound guy. So anyway, back to this. This is the glycoprotein structure, or it's not necessarily the glycoprotein structure, but it's the binding protein structures. I think this one here that he's talking about is the influenza, and this little sugar molecule. Yeah, it's a little binding receptor. So yeah, it's a lot. I know. Actually, you know what? Let's take a little break from the science, and let's go look at a very cool video about what it looks like computer generated on a cellular level. And the best one I found was in TED Talks. I'll never find it now. CRISPR virus hijackers. Just one sec, bear with me. Oh my goodness. Yeah, it's just a mess trying to find something in all this information, and my apologies. Okay, I'll find it. Just give me a second. It's gonna be forever. Actually, wait. Where's my like library stuff? Oh, maybe I go over. Yeah, we'll go back here. Here we go. Again, my apologies for trying. I'll just try to get stuff together and get it out in such a way. Where is it? TED Talks. Is that it? Oh my gosh. Of course, I'll never find it. But anyways, here is actually it's not the one I was looking for, but let's go in to see how the inner life of a cell. This one's pretty cool. I don't want to answer questions right now. Wasted enough time already. All right. Let's kind of give you a visualization of what's happening, right? Okay, I'm just gonna stop this here for a second. Like, now picture like the ocean, right? Like the ocean and like the little sea creatures that live in the ocean. Now, picture like sitting 1,000 light years away somewhere on a distant planet and like trying to look in our ocean and see those little creatures in there doing what they're doing in our ocean. Alright, it's almost like the same kind of perspective as what we're looking at down in we're looking so far down. We're looking at a level that's like all this empty space that we're seeing here. Like that would be like, let's say like protoplasm or like whatever liquid that's suspending all this happening, because this is beyond like our this is beyond a smaller level than even our blood cells. Like way down. So just keep that in mind too, right? It's perspective, and the more that we can kind of visualize what's happening, I think the more we're going to get an understanding of why these proteins are so vital in what they do, and why they're such a key point in this specific strain, and why a lot of speculation is circulating. Now, I want to pause this here for a second, because I know this probably doesn't even seem like it's real. This thing walking along a path, and it's carrying... What's it carrying? Well, that's carrying a lot of information. And it's going up the informational pathway, traveling along our informational highway at a genetic level. And to understand this makes it a little bit easier to kind of visualize the scientific words. Again, yeah, this actually is a process that happens, and it looks like it's walking. And the reason why it's walking is because it's static electrochemical, and like it's fucking far out there, I promise you. But you don't have to understand every little thing, but as long as you can kind of get a visual representation, you're going to understand why this is so important, why these proteins play such a crucial role, why every little specific tiny little detail in the slightest variation can have the biggest impacts on anything. what's happening right there is kind of like the mechanism that creates our DNA. And this thing is copying it, making more copies. It's the mechanism that controls this. So it's all these big strands. It's all genetic material, amino acids, like fucking proteins. It's like just basically big chains, and it's copying these chains, and it's crazy. It really is. It's cool though in a sense, but you know, at the same time, there's a lot of vulnerabilities at this level, right? Especially if you have something that's designed to go after your vulnerable aspects. What just happened there, that kind of transition or that fusion kind of motion that just happened there, that's basically what's happening with this specific type of virus. It's using fusion, so it kind of just like blends right in with our virus and leaves like encapsulated. I can't even shut up about it because it's just so much information, but I think that's basically that's actually probably good for that video. But it gives you an information or a visualization of what's happening on these microscopic levels, so to kind of, you know, put into perspective what this guy's doing. Like, I know it just looks like little connecting lines and different colors and stuff, but all these different colors are like different proteins, amino acids, different structures, and all those things we've seen in the previous videos. Those little jiggly wiggly things, like, it's crazy what makes up our biology, and you know, specific things that are tuned to take advantage of our weaknesses are not good. So it's definitely pointing in that direction though. Yeah, it's just too much to even anyway, back to the video.
And so let me show you what the Wuhan coronavirus is recognizing, and also SARS, a virus that caused an epidemic in 2003, what it recognized. Oops. Okay, so let me try to get it. Where is it? Here it is. So Wuhan virus and SARS virus are recognizing this protein, which is, again, it's a natural protein in your body, like this sialic acid on the surface of the cells. This is called angiotensin converting enzyme 2, ACE2. There's also an angiotensin converting enzyme without the 2. Okay, this one with the 2 lowers your blood pressure. The one without the 2 raises your blood pressure. Both of them work. They have this groove.
And just right there, if anybody wants to go look at the case studies, you'll notice that all the patients had low blood pressure when they have this. So it is, you know, it all connects, right? This ACE2 and ACE1, the one will increase your blood pressure, and ACE2 will decrease your blood pressure, just because of little snippets of you know, these little particles, right, off the genome, and it all just two little particles in the difference creates either up or down blood pressure. You know what I mean? So that's how sensitive this stuff is, right? And you got it's there's big differences in I'm trying to show you just how even the small differences make the biggest differences sometimes at this level.
Let's see if we make this groove bigger. This groove here, right in here. Both of them work by clipping a small angiotensin molecule. It's 10 amino acids long. The ACE without the 2 clips it from length 10 amino acids, angiotensin from 10 to 8, and that raises your blood pressure. And this ACE2 clips it from 8 to 7, and that lowers your blood pressure. So this is actually a good molecule. Both ACE and ACE2 are interesting targets for a lot of diseases, for high blood pressure, for diabetes, for heart disease. At any rate, we're not going to be.
But what he was kind of saying there, diabetes, heart disease, not necessarily viruses are going to target this region. Basically, what he's saying there. He said, it's not a region where a virus would normally target, and because of that, it's highly, strange.
Be able to avoid Wuhan coronavirus by getting rid of this. This is an important, good molecule on the surface of your cells. So, how does the virus spike recognize this molecule? Let me bring up. I'd like to show you the Wuhan coronavirus, but we don't have a structure of it. So I'm going to show you SARS, which is the closest thing we have, the spike structure of SARS. Okay. And this is going to look like a lot like that human coronavirus, still the 3 proteins. The sugars aren't here, but they aren't shown here, but in fact, there are sugars here. It was just in this particular model, they didn't add the sugars. Those sugars are part of all the spikes, the coronavirus spikes. And what they saw when they looked at SARS is that these, at the top of the spike, the virus would flip out part of it would make a small motion. Let me show you what that motion looks like if I can. So let's see, here we go. Okay, do you see this little green part at the top? It's called the binding domain. And it can move from a down position where all 3 of them are down and kind of tucked in to an up position. And that up position is the only position that can recognize this ACE2 molecule, the angiotensin converting enzyme 2. When they saw this in electron microscopy, they only would see 1 up at a time. Like the red, blue, and green are identical, but only 1 would be up at a time. They didn't see 2 up, they didn't see 3 up. They just sometimes see 1 up, sometimes see it down. Okay. When it's up, they also saw it bound to this ACE2. And it doesn't bind to just any old place on the structure. It's very specific on where it wants to stick to the ACE2. So let me show you exactly the correct location where it sticks. Okay, so here's the actually from the electron microscopy that shows the ACE2 in brown. Here's the spike, it's flipped up. When it's attached to the ACE2, what they see in the electron microscope is the thing is even more flexible. So let's see. There we go. Let me see if I give you a better angle that shows this motion. Okay, so the thing can swing in and out. And what they observe next is that half of this spike just falls off when the ACE2 attaches. This is in SARS. Okay, what the top half falls off. And here I changed the color scheme on you a little bit. The bottom half I colored light colors, light red and light blue, and this light green kind of looks gray. The each of these 3 copies of the protein is cut at a certain point, and I marked it in red where it's cut. A protease, another molecule, makes a cut right here, and that's totally critical. If the cut's not made, then the virus can't infect. Okay, so each of the 3 ones has a cutting point, and what they observe is this green copy, the top half with the ACE2 floats away, and then the red and the blue, they don't have ACE2, only one of them. These become unstable. They no longer stay attached, and they float away, and we're left with just this bottom light-colored portion of the spike. And this bottom portion in the middle exposes the mechanism which allows it to fuse with the human cell. Okay, is this what's going on in Wuhan coronavirus? So everything I'm showing you is for SARS. We don't have the structures for Wuhan coronavirus, but one thing we can do is we can look at the difference between this SARS virus and Wuhan virus, because we know the chemical structure of the Wuhan virus from its genetic code. So let me color the differences between the two. Let's see, I have a little button I made to do that. There we go. So I colored every part of this green molecule, 1/3 of the spike, orange if it was different between the Wuhan and the SARS virus. So you see, it looks like a lot of orange. It's only about.
Yeah. And what he's talking about, like, this is big differences. Huge differences. Why does that matter? Because this is the binding sites. This is the communication sites that allow you to communicate with the virus to infect it and so forth. Communicate with the ACE2 receptor. It's showing some very highly, and especially up here, right? These are the connecting points, right? This is where the glycoprotein 120 is, and here, and the amino acids, where these connecting points are. And that's what the same thing is what they're associating with this paper that everybody blew up about. No, it's not there. No, you guys are crazy. No. Fake news, paper's fake, right? But it's the glycoprotein, and what does it do? It has a high affinity binding site, the co-receptor CXCR4 CR5. I know that might sciencey stuff. That's what these binding proteins. That's what it's activating, and it's activating from our T cells. Our CD4 cells. Right? I don't know a whole lot about it, but I know that everybody's scientific analysis now is kind of supporting general theory that spike proteins are showing some very odd, definitely some very odd characteristics, and they shouldn't be there. Like, the chances of them forming the way they did and where they did is just too the high. Probabilities asterisk. Back. Yeah. We don't really need to actually go to that video. Because basically he's just going to keep going into the structure. Actually, yeah, we will, because he is talking about the differences right there.
25%, it's about 75% identical, which is a high amount of identity. When 2 proteins in your body are 75% identical, generally they function nearly exactly the same. Okay, but it would be what we'd really want to look at here is sort of the really important part here, where it's attaching to this ACE2 molecule. We see a lot of orange at the
A lot of orange. Why would we see a lot of orange at the receptor binding site? I got a better. Okay, so this specific glycoprotein is cleaved into 2 subunits S, and to help the receptor binding and S2 to facilitate the membrane. The spike glycoproteins of coronaviruses are important determinants of tissue tropism host range, meaning basically that these proteins are responsible for controlling or regulating what virus or what cells they can, what range of species. On careful examination of the sequence alignment, we found that the glycoprotein insert, they are, unique. This so these binding sites is basically how it's going to be able to recognize the cell, infect the cell, right, through these binding sites. And if binding sites are enhanced in some kind of way, it would make it more infectious in turn, right? Of course it would. But it's not just, you just can't pick and choose little things and make it work. This is, highly skilled level to do this kind of thing. Back into this and the protein talking about up here in the orange. This is the binding site. That there's a lot, differences with this between the other ones. Binding site has something definitely worth notably. Okay, so anyway, let's go.
Interface between the two. And that would make me suspect that this isn't quite right. Wuhan virus. SARS virus and Wuhan virus are probably a bit different here. And in fact, if I only had this evidence, I would probably think that Wuhan virus wasn't even using this ACE2, okay, because there are so many differences right there. But experiments that were done in the last few weeks already confirmed that Wuhan virus indeed is using ACE2, okay? But we can expect that our model based on SARS is going to be a little bit off because there are a lot of differences in the critical location where it binds.
A lot of differences. The location where it binds. Surface proteins. Glycoprotein 120, whichever. All know these HIV-1 proteins help. And wow. I must note, although I'm not a scientist, a little bit, but the PI values are 10, negative 8, 12. There's a variable in there. Those values are extraordinarily high. What do those mean? I don't exactly. Wish I could tell you. I know it does affect how infectious. That's a high number, considering most of the things are around 4, 5, 3. And evolutionary analysis from this report. It's been speculated this virus is a variant of coronaviruses derived from an got transmitted to. It's been speculated, yet. Considering the chance of specificity for host means considering the chance that this is referring to spread through humans, we decided to study the sequence of the virus. S proteins are surface proteins. Help with virus and host recognition attachment. Excuse me. Thus, a change in these proteins can be reflected as a change in meaning that if people change these proteins in a certain way, they could in fact make this spread through bats, because bats are usually of these types of coronavirus. Usually spreads through bats. I'm just coughing up a storm. It's just because I'm talking. So yeah, these proteins are very quick and they allow the communication fire even better. Of note, all 4 inserts have PI values of 10, may facilitate our findings suggest unconventional evolution. Warrants further investigation. With that being said, I can't stop coughing. Woo. So, warrants further investigation. I will do that. I hope you do that. Bring you the latest. Oh, my. Thanks for watching. Hope you got something.